on the myelinating cells in my lab and how to modulate their plasticity to enhance the axonal regrowth and remyelination. So, of course, you must be all aware of that. I just do a little introduction in case there are some students coming. So the myelinating cells, they're two types. They're oligodendrocytes in the CNS and Schwann cells in the PNS, and they're very different. They originate from different progenitors, and you can see on this drawing, they form quite a different myelin. So they are very different. However, they share some common function. They provide axonal insulation and the saltatorian of conduction. And this myelin that they form is critical for the functioning of the nervous system, because if there is no myelin, then the nervous system is poorly functional and is very sensitive to degeneration. So in humans, there are different cases where there is a demyelination or dysmyelination, so aberrant myelin formation. So this can be due to autoimmune and inherited demyelinating diseases. And I'm going to cite a few. Of course, this is not exhaustive list. There's a long list of diseases, but the most frequent or the most famous in the central nervous system, multiple sclerosis. So it has a strong immune component. And it's very frequent. It touches the young adults between 20 and 40 years of age. So it's and then of course we have no cure. So it's a very important disease to work on. Leukodystrophy, so it's a large group of inherited developmental hypomyelinating diseases. Schizophrenia, we know since some years now that it is linked to demyelination, or at least some subtypes are linked to demyelination. And in the peripheral nervous system, there's a large group of peripheral neuropathy called Charcot-Marie-Tooth diseases that are inherited, and some subtypes are demyelinating. In the case of the Guillain-Barré syndrome, there's demyelination that is induced by a viral infection, and in some cases, people can fully recover, but in other cases, there's some defect that remains. And neurofibromatosis is a tumor formation that can lead to demyelination. Then demyelination can also be secondary to diabetes or toxic agents. And during aging, there's always some demyelination, more or less, depending on individuals. And of course, after trauma, demyelination occurs. And in the case of the central nervous system, the lesions do not regenerate or very poorly. So there's not a good regeneration. And in the PNS, in contrast, there's a very good regeneration. So in my lab, we are very interested in traumatic lesion. So how to modulate the system, understand how the PNS regenerate, and try to apply what we learn to the CNS to promote regeneration. We work also on multiple sclerosis and also a little bit more in collaboration on Charcot-Marie-Tooth diseases. So I'm going to talk right now about the lesion. So how does it work? How does the PNS regenerate? So when there is a lesion in the adult peripheral nerve, the Schwann cells, so the myelinating cells in the PNS can sense this lesion, and they react to that by demyelinating. So they actively lose their myelin, they digest it. So that's a process called myelinophagy. And then they convert into repair Schwann cells. And those repair Schwann cells, they stimulate axonal regrowth, and then they guide axons back to their former targets, which is very important for functional recovery. And once axons have regrown, then Schwann cells redifferentiate to reproduce the myelin around the regenerated axons. So we can have almost full regeneration in the peripheral nervous system due to this high plasticity of Schwann cells. But in contrast, oligodendrocytes, in case of a lesion in the CNS, they cannot do this reaction. So they do not demyelinate at all. They actually continue to try to myelinate the damaged axons, or they die. And in any case, their myelin is not removed, and it contains several growth inhibitory factors for axons. So the axonal regrowth is blocked, is prevented by the myelin of those oligodendrocytes. So this is one of the main reasons why the CNS does not regenerate efficiently, whereas the PNS can regenerate very well. So we are very interested in my lab to understand the mechanisms that control the different plasticity of the Schwann cells and the oligodendrocyte. And so to modulate this plasticity and to test what is the effect of this modulation on axonal regrowth, we have set up some lesion models. This was the work of Adrien Vaquier, a very excellent PhD student in my group. So we use a classical microfluidic chamber with two chambers that are connected by microgrooves. And in one chamber, we plate a primary neuron. So we mostly use DRG neurons because they have long axons. So these neurons have big cell bodies, so they remain in this chamber. The cell bodies remain in this chamber, but their axons, they grow through microgrooves and they invade the second chamber. And so in the second chamber, we have myelinating cells, so either Schwann cells or oligodendrocyte. And the Schwann cells, they can also interact with axons in the microgroove fully, so they go fully in the microgroove, whereas oligodendrocyte, they do not go fully, but they also myelinate either in the microgroove or in the chamber. So, so Adrien let this system grow at a more or less stable stage, most stable as possible, that is myelinated. And then what he did some axotomy by laser. So this system is coupled to a spinning disc, so where we can do very fast lesions. So you have to imagine that in this model, we have about 200 microgrooves. So you need to be very fast to make sure that you have synchronization. So the spinning disc allows to be very fast. And what he did also, he used lentiviruses to label the neurons in red and the myelinating cells in green. So we can do with this system some live cell imaging, time-lapse imaging, and then we can follow axonal regrowth. We can follow the interaction between the two cell types. So it's really a very nice system to test different hypotheses. So if we look into this microgroove, that's what we will see. So you can see here an example with some red axons and some green Schwann cells on the top. So this is live. Huh? And here you see an example of the system with the oligodendrocyte. So you can see the oligodendrocyte. So they are here, they stick to the wall of the microgrooves, and then they send processes in different microgrooves to myelinate different axons. So that's what they do. So what is really amazing with this system is that they really mimic what happens in vivo. And if you look at the model with Schwann cells, you can see here this example. So you can see this red axon here, and on the top of it, you can follow this Schwann cell that has a typical morphology of a myelinating Schwann cell. So when you do a lesion, very rapidly, you can see here already one day post lesion, the axon is totally disintegrated, and you can see the change of morphology of the Schwann cell. And then later on, the Schwann cell extend a long process, so it's not extremely visible here, but we see it a bit later, extend a long process to the tip of to the growth cone, to the tip of the axon, and the axon uses this process to regrow on it. So you can see it uses the process, and at some point the Schwann cells cuts out the process, reinteract with the axon, and then remyelinates it. So in this model, with only neurons and Schwann cells, you can have all the steps of the regeneration process that you can see in vivo with a timing that is extremely similar to what happens in vivo. So we use a lot this model to study the different steps of the regeneration process. And what I can show you also, what we can do so we can do movies. So for example, here, those movies have been taken 20 minutes after lesion. So the lesion is on that side here. So I show you two movies, and those are the Schwann cells. So you can see very fast after lesion, axons disintegrate, and then the Schwann cells, they move towards the regrowing axons. And if you look carefully, you can see that the axons will regrow, follow the Schwann cell and regrow on the Schwann cell that move towards the axon. So it's quite beautiful, quite amazing to see that. I mean, that's what they're supposed to do, but they really do it also in this model. I can show you a second movie here. So a different groove and a bigger magnification. So you can see the axon is disintegrated, and the Schwann cell will extend a very long process. This one was really amazing, extends very long process. And as soon as an axon has made contact, then it really follows the Schwann cells to regrow. So you can look at axonal guidance with this model. You can measure axonal regrowth. What we also did with the spinning disc, so you can image very fast. So we did time-lapse imaging, but also confocal imaging, and we reconstructed 3D movies. So I will show you some example here. So in that case, you can see the red axons and the green Schwann cell, and the lesion has been done on that side. And for this movie, it has been done on that side. So I'll show you how it works. So very fast after lesion, the Schwann cell moves really fast around the axons. And what we had the impression when we saw this movie, so those were our first 3D movies, that the Schwann cells was promoting the disintegration of axons. I can show you another movie here. Yeah, so here I picked two key images from this movie. So you can see here the Schwann cells enwrap the axon, and 10 minutes later the axon is disintegrated, and here the same Schwann cells enwrap the axon, and then it disintegrates, the axon is disintegrated 10 minutes after. So this is actually what we have shown in this article that was published last year in Cell Report. We show that shortly after lesion, the Schwann cells accelerate the disintegration of the distal part of the axon that is cut out from the cell body. And this accelerates axonal debris clearance and also accelerate axonal regrowth. And so Adrian has shown very nicely that, that the Schwann cells, very fast after lesion, they polymerize actin. You can see those actin spheres that are polymerized after lesion. Those are different examples. And you can see that in the spheres, this is neurofilament, this is phalloidin that stains F-actin. And you can see in the spheres, the axons get swollen. You can see it depends at which stage we are. So that's a different stage. It has swollen also. You can see the constriction at the contact of the actin spheres. And here it's different stage, a later stage. Here you can see it's totally disintegrated and you have the sphere here. And that's a nice 3D reconstruction. So very nice sphere. You can see the swollen axon here. And this one is also very nice because you can see that the axon is constricted at the point of contact with the actin. And at this stage here, the axon is totally disintegrated and the part that is disintegrated remained in the sphere. So as Jorge said, so we found that this mechanism was a cross talk between the distal cut axon that expressed that did the local translation induced by injury of PLGF, which is an agonist of the VEGF receptor 1. This induced the activation of the VEGF receptor 1 in Schwann cell and its upregulation. And this led to the polymerization of actin, the formation of these actin spheres. And the oligodendrocytes are not capable of doing that, so they do not polymerize actin. However, when we supply them with lentivirus, we gave them VEGF receptor 1, so they could express VEGF receptor 1, then they were able to polymerize actin. So they didn't form those spheres, so they didn't have the same morphology, but they formed small arms of actin along the distal cut axon, and they could also accelerate the disintegration. So that was really quite amazing that oligodendrocyte can acquire similar behavior as Schwann cells with only one protein. So we were really about this study. I thought it was really amazing to see. So what I want to show you also what we can do with this model. So you can do RNA-seq and you need only one chamber. You have enough material with one chamber to do RNA-seq. So that's what we did. We took the content of the the myelinating cell chamber, so either Schwann cell or oligodendrocyte. And what we compare is the gene regulation at 1 day post-lesion as compared to no lesion. So we found many genes regulated in both situations. And if you do the gene ontology analysis, what you can see basically is that oligodendrocytes react oppositely to Schwann cells when you do a lesion. So I'm not going to go through everything, but this is quite nice because that's what we would expect actually, but still it was nice to see, and the information is also great. And what we found, which we are very interested in, we found 7 genes that are oppositely regulated in Schwann cells and oligodendrocytes. So right now we are working on these genes. We have started to work on these genes to see whether, if we revert the regulation of these genes, whether we could, in oligodendrocyte, whether we could revert or we could gain the same behavior as Schwann cells. So yeah, that's what we're doing right now. And then, as Jorge said, we are actually very much specialized. So that the second part of our work is the work on chromatin remodeling, which is actually the main part. So we work a lot with chromatin remodeling enzymes of the histone deacetylase and also histone demethylases family. So chromatin can be remodeled by different modification, either directly on the DNA by methylation or on the histones by various modifications. And so all those modifications, they will modify the compaction of the chromatin. And by doing so, they modify the accessibility of the gene. So they can, if the chromatin is decompacted, then the accessibility for the transcriptional machinery is better than, of course, if it's compacted and inaccessible. So we work on that, and mostly we work on acetylation and methylation, but of histones, but there are actually many post-translational modifications possible on histone. They can be phosphorylated, ubiquitinated, sumoylated, ADP ribosylated, and many other modifications are possible. And potentially all these modifications, they can change the architecture of chromatin, and then the gene accessibility, and then the gene activity. So if we look at since I'm going to present some work on histone deacetylase 1 and 2, I will tell you how it works, histone acetylation and deacetylation. So when histones are acetylated by histone acetyltransferases, their attraction to the DNA is loosened. So this results in a relaxed chromatin that facilitates the access for transcription factor and the transcriptional machinery. And when instead histones are deacetylated by histone deacetylases or HDACs, then the attraction to the DNA is tight, and this leads to a locally more condensed chromatin that will limit or select the access for the transcriptional machinery. So those HAT and HDAC, they control transcriptional activity at the chromatin level, but they also have a lot of non-histone targets. And among those, many transcription factors. So they can also control transcriptional activity by modulating the activity of transcription factors. So they are very powerful transcriptional regulators. So there are several families or classes of HAT and HDACs, and in my lab, we are mostly interested in the class 1 HDAC. So this comprises HDAC 1, 2, 3, and 8. And we're interested in those HDACs because we have seen that they're highly expressed in Schwann cells during development, but they also strongly regulated after lesion, after a peripheral nervous system lesion. And so I will, we have mostly work on HDAC 1 and HDAC 2, so I'm going to present, where we were working on HDAC 3, but then our competitors published before, so yeah, that's the way it goes. So I will talk about HDAC 1 and HDAC 2. So what we found is that those two HDAC play a key role in Schwann cells during PNS development, maintenance, and remyelination. And if you study HDAC 1 or HDAC 2, you usually have to study both together because they are highly homologous and they can usually compensate for the loss of each other. So it's very difficult to work only on one. So we found that those two HDACs are critical for the specification of neurocrest cells into the Schwann cell lineage. we found that they're also essential later in the development for Schwann cell myelination and survival, that they maintain paranodal and nodal integrity in the adult PNS. And a very interesting study, we found that actually the system is not perfect, that HDAC2, in case of a lesion in the peripheral nervous system, it induces the formation of a multi-protein complex that, that includes other chromatin remodeling enzymes, so H3K9 demethylases. And this protein complex also includes SOX10. So this transcription factor, as you all know, I'm sure that is essential for the life of a Schwann cell, for this differentiation process and for myelination. So this complex is formed too early after lesion. It's formed very early after lesion, and it slows down axonal regrowth. But later on, it is necessary for remyelination. So what was really interesting here is that we could use an inhibitor of HDAC2 and HDAC1, and this blocked or delayed the formation of this complex. So we could promote axonal regeneration, axonal regrowth. So we had faster axonal regrowth, and then we stopped the inhibitor of HDAC early enough to not impair the remyelination. So in the end, we had more efficient axonal regrowth and even more efficient remyelination, because probably the remyelination started earlier than in the control animals. So in all these studies, so we found mechanism of action, but what we were really missing was to know what is the direct target, deacetylation target of HDAC1/2 that will allow the function on the Schwann cell differentiation process and myelination process and remyelination process, because each time we found a mechanism, each time it was linked to SOX10. So we thought, so what is this deacetylation target of HDAC1/2 that allows the activity of SOX10? So this is the topic of our latest publication. So we published that a couple of months ago in Nature Communications. So we found that when we increase the activity of HDAC2 and its expression, we can enhance remyelination efficiency in the PNS after traumatic lesion, but also in the CNS after demyelinating lesion in young and old adults. So the first aim of these studies was to find this famous deacetylation target of HDAC1/2. So to do that, so this was the work of Mert Duman, another PhD student in my group, also very talented. So what he did, he treated mice with an HDAC1/2 inhibitor called mosetinostat, or with his vehicle. Then he did a sciatic nerve crush lesion, and he collected the nerves at 1 day post-lesion, where we know that this multi-protein complex dependent on HDAC2 is already formed. And then he did an immunoprecipitation of acetyl-lysine, and he sent that to mass spec. So what we were expecting to get as a result was that, okay, if we were expecting to have, if there's a direct deacetylation target of HDAC1/2, we're expecting to have more in the IP on the nerve of the mice treated with the HDAC1/2 inhibitor. So we got actually 2 potentially interesting targets that we judged potentially interesting, and then we did an IP together with HDAC2, and we found that among those 2, eukaryotic elongation factor 1A1 was interacting with HDAC2. So we followed this target, and we could confirm that HDAC1/2 deacetylate this EF1A1 after lesion. So we wanted to know whether it's also a target of HDAC1/2 during development. So to answer this question, we knocked out HDAC1 and HDAC2 together. So we crossed HDAC1, HDAC2 flux mice together with mice expressing the Cray recombinase under the deserted shock Cray, onto the deserted shock promoter that is active during embryonic life in Schwann cells, but after specification. So what we found is that there was a huge increase of the level of acetylated EF1A. So there's an antibody for that detects the levels of acetylated EF1A. Huge increase of acetylated EF1A in mouse pups at P4, 4 days after birth, as compared to control where the levels were almost not detectable. And you can see here, this is the IP of EF1A1, and then the blood for acetylated EF1A. So then we concluded that HDAC1 deacetylate EF1A1 also during development. So what is EF1A1? So that's a major translation elongation factor, but it also has a non-canonical function. So it has been described to be, so as translation elongation factor is mostly localized in the cytoplasm, but it has been shown to be also localized sometimes in the nucleus and to regulate transcriptional activity, nuclear export of tRNAs or apoptosis, so different functions. So we went on with that target. So we did some purified primary Schwann cell culture, and those cultures, we can culture them in differentiating conditions that mimic pretty much the state of a mature nerve, mature peripheral nerve, or we can also de-differentiate them, and this mimics also very well the state of Schwann cells early after lesion. So what we saw that there was an increase of acetylated EF1A levels in de-differentiating conditions in the Schwann cells. And because we were looking for a direct target of HDAC2 that regulates SOX10 activity, we also looked at SOX10. What we saw is that in de-differentiated Schwann cells, you have relocalization of a part of SOX10 in the cytoplasm, and you have decreased SOX10 level. And you can see compared to the differentiated state, SOX10 is very much nuclear. So we were looking whether there is a potential link between those two events. So what we did, we kept on with the primary Schwann cell culture under de-differentiating conditions. We IP SOX10. We separated the nuclear fraction and the cytoplasmic fraction first. We IP SOX10, and we found that SOX10, that acetylated EF1A, co-immunoprecipitate with SOX10. And so when we treat the cells with the HDAC1/2 inhibitor mosetinostat, you can see the levels of acetylated EF1A are very strong, very much increased. And acetylated EF1A is localized in the nucleus, but also in the cytoplasm in both compartments. So this treatment with mosetinostat increases even further the immunoprecipitation, the interaction between acetylated EF1A and SOX10. You can see the SOX10 levels are very low in the nuclear fraction, but still you have more interaction. So we also checked whether HDAC2 was co-immunoprecipitating. So that was the case only in the nuclear fraction. So then we had this confirmation that acetylated EF1A interacts with SOX10 and HDAC2 in the differentiated Schwann cells. So then we wanted to check that in vivo. So we checked by immunoprecipitation whether EF1A1 was really acetylated after lesion, so 1 day post lesion. So we did here, this is a crush nerve, and that's the contralateral nerve of the same mouse. So it's a good system because we can use an internal control for each mouse. So we did an IP of EF1A1 and blotted for acetylated EF1A. In the end is acetylated EF1A1. So you can see that there's an increase already at 1 day post lesion. And this correlates very well with a strong decrease of the SOX10 level in these nerves at 1 day post lesion. So then we wanted to know, actually it was already published, but we wanted to double check that, whether Schwann cells or whether SOX10 is degraded in the proteasome, and whether after lesion or in a de-differentiated Schwann cell that would mimic a lesion, whether SOX10 would be degraded in the proteasome. So to look at that, we did co-immunofluorescence of SOX10 with the 20th subunit of the proteasomes, and we could see indeed that SOX10 some of the SOX10 localized in the cytoplasm was enwrapped by the proteasome. So you can see here the 20S by 3D view. And we treated with the proteasome inhibitor only shortly, only 12 hours, because the cells don't like it so much. And you can see already 12 hours with this treatment, you can see an increase of SOX10 levels. So after, at least in dedifferentiated Schwann cells, SOX10 is targeted to the proteasome. So then the next question was, so does EF1A1 target SOX10 to the proteasome? So does it induce the relocalization of SOX10 in the cytoplasm and then induce the degradation? So to look at that, we transfected Schwann cells with EF1A1. So we overexpress EF1A1. And one day post-transfection, you can see that SOX10 in the cytoplasmic compartment is more abundant and is less abundant in the nuclear compartment. And that's the quantification here. And if you wait longer, so 3 days post transfection with EF1A1, you can see here the transfected cells. And in most of these cells, you can see the levels of SOX10 are extremely low. And this is the control GFP. You can see that in contrast, the levels of SOX10 remain mostly the same. So if you increase the expression of EF1A1, you relocalize SOX10 into the cytoplasm, and then you decrease the levels of SOX10. So here, so we also did work with mutant acetylation mutant. So we wanted to know whether there are going to be some potential acetylation sites that are very important for EF1A1 to interact with SOX10 and then induce its degradation, and also to go to the nuclear compartment. So as you can see here, we were quite, we were quite desperate when we saw the molecule. So it's highly post-translationally modified, and there are 20 potential acetylation site. So it's mostly Mert was desperate, and I was I was sorry for him. So what we did, we first looked at the literature, and we decided on 3 lysine where we thought, well, okay, those lysines have been experimentally verified that they were acetylated and published several times. So we chose lysine 41, lysine 179, and lysine 273. So the one additional advantage of the lysine 273 is that it's specific to EF1A1. So there's a quite homologous protein that is EF1A2, but this one, instead of a lysine, has an arginine, so it cannot be acetylated there. So it was quite interesting for us to test this acetylation site. So what we found, I'm not going to show you all the experimental details. So what we found when we mutated the lysine into glutamine that mimics acetylation, we can see that any mutant lysine to glutamine actually had an increased nuclear localization. So any acetylation site led to more nuclear localization of EF1A1 and also decreased the levels of SOX10. Then when we did the opposite, so we mutated the lysine into arginine to prevent acetylation, then we could see that the lysine that were the most efficient to maintain EF1A1 in the cytoplasm and to prevent the decrease of SOX10 were lysine 273 and lysine 179. So now I cannot tell you if other lysines are important, but at least those seem to be very important. And we could confirm also this lysine by mass spec. And this lysine here, this acetylation of this lysine is the antibody we use. So we know that it also gets acetylated in vivo and in Schwann cell culture. So I don't have time to show you all the experiments, but that's the mechanism of action we found. So we found that shortly after lesion, TIP60, which is a histone acetyltransferase, coordinates its action with STAT3 to acetylate EF1A1. acetylated EF1A1 then translocates to the nucleus. It interacts with SOX10 and then drags SOX10 out of the nucleus and targets it to the proteasome for degradation. And then conversely, HDAC2 deacetylates EF1A1 to send it back to the cytoplasm and then maintain SOX10 on its target gene, and this can allow remyelination. So we were very happy with this mechanism, but then we wanted to use it potentially to enhance remyelination. So we thought, well, very naturally, well, let's try to increase the activity of or expression of HDAC2 and then get rid of this acetylated EF1A1 and allow the remyelination by SOX10. So what we did, we looked for an activator of HDAC2, and there's actually only one known. It's called theophylline. It's already used in asthma or COPD, so chronic obstructive pulmonary disease, but it's a very old drug and it's mostly not used anymore. Other drugs have replaced theophylline, but at a low level, low concentration, theophylline is a potent activator of HDAC2, so it increased the activity and also the expression of HDAC2. And at higher dosage, then it's an inhibitor of phosphodiesterases and is also an antagonist of adenosine receptors. But there's a big difference of concentration between those targets. So we could have a low dosage of theophylline to act on HDAC2. So what we did, we treated the mice after sciatic nerve crush lesion at 10 days post-lesion, or I think 9 days, I don't remember what we did, I think 9 or 10 days. So just before the Schwann cells start to remyelinate, and we treated either for 2 days or for 4 days. And then we looked at what happened afterwards. So you can see that already 2 days of treatment, so 12 days post-lesion, you have, so it's not very visible, but there's already, so theophylline increased the activity also of HDAC2, not only its expression. So, but there's already significant increase of HDAC2. You can see it better after 4 days of treatment. There's increase of SOX10 at already 2 days, also at 4 days post-treatment, increase of CROX20 and of P0 at all time points. And even at 30 days post-lesion, even if you treat only 4 days, you still have a strong increase of CROX20 and P0, myelin protein 0, which is one of the main myelin protein in the PNS, and CROX20 is a major transcription factor for myelination. So we can increase very efficiently the expression of promyelinating factors and myelin proteins with theophylline. We show that after 2 days of treatment, you can see the level of acetylated EF1A here in a vehicle-treated mice. And in the theophylline-treated mice, you can see the levels are very, very low. So we show that by immunofluorescence, but also by immunoprecipitation. So you can see the crush nerve here, the contralateral nerve of the theophylline-treated mouse, and that's the crushed and contralateral of the vehicle-treated mice. So you see the levels are very low, and this is quantified here. So theophylline decreases the level of acetylated EF1A1. So what about the morphology, the myelin sheaths? So what we could find also at 4 days post-lesion, we can see already, so those are cross-section of sciatic nerve. And this is electron microscopy pictures. You can see this clear round shapes are axons, and around it, the dark ring is the myelin sheath. And you can see that this myelin sheath is already thicker after 4 days of theophylline treatment. And this is quantified here by the G ratio. So the G ratio is the ratio between the diameter of the axon divided by the diameter of the axon plus the myelin. So the lower the G ratio is, the thicker the myelin is. And this G ratio is extremely constant. So a small difference makes a big difference actually in the myelin thickness. So for 14 days post-lesion, 4 days post-treatment, you can see already a thicker myelin, and this advantage is still visible at 30 days post-lesion. You can still see there's still a little difference between the theophylline treated mice and the vehicle. Of course, the vehicle treated mice also regenerate, so it's just faster. However, what we could find, so we checked at 2 months post-lesion, and what happens when you do a lesion, you have remyelination, but the myelin thickness will always remain thinner as before a lesion, or as compared to an animal that didn't have a lesion. But what we could achieve with theophylline is that the thickness of the myelin was comparable, similar to a non-lesion nerve. So we could recover the same thickness of myelin. So then we did also functional tests to see whether the animals could recover their functional, so motor function and sensory function faster with theophylline treatment. So you will see here a video with of animals treated for 4 days with theophylline, and this is the control group treated with vehicle. So, and this is rota rod. So we put the animals at a constant speed on this rota rod. And at 14 days post-lesion, they are not capable of staying very long on the rota rod. They are not yet very fit. But the group treated with theophylline is very fit. So those animals can stay much longer on the rota rod. So they recover functional motor function faster than the vehicle-treated group, but at 30 days post-lesion, we should have done something in between. 30 days post-lesion, the vehicle group has recovered also and is capable of doing the test. So we don't see a difference anymore. However, for the sensory test, so we did a toe pinch test, and here we can see the advantage of the theophylline treatment at 14 days post-lesion, but also at 30 days post-lesion. So the animals recover faster and more efficiently. As we could see with the electron microscopy. So yeah, so that was great. We were very happy. And then we were wondering next, could we use theophylline to increase remyelination efficiency in the central nervous system? So to answer this question first, we check whether HDAC1/2 were also controlling the deacetylation of VF1A1 in the central nervous system. So we treated mice with the HDAC1/2 inhibitor mosetinostat intrathecally and collected the spinal cord 1 day post-lesion. And here again, we saw a really strong increase of the levels of acetylated DF1A that were not detectable in the vehicle-treated animals. And you can see that that's a co-staining with CC1, which is a marker of mature oligodendrocyte. And you can see that this oligodendrocyte, I mean, acetylated DF1A is everywhere, but it is strongly also localized in the oligodendrocyte. So, okay, it seems that HDAC1/2 activity is also necessary in oligodendrocyte to deacetylate DF1A. So we did primary oligodendrocyte cultures, we differentiated them, and then we treated them for 24 hours with the HDAC1/2 inhibitor mosetinostat. And you can see here, that's the vehicle-treated cells and the mosetinostat-treated cells. You can see the strong increase of acetylated DF1A in the CC1 positive cells. And very amazingly, so that was really strong effect. You looked at the SOX10 level in the vehicle-treated cells as compared to the mosetinostat-treated cells. So basically, SOX10 is totally relocalized to the cytoplasm, almost totally, and the levels are very low. So that's a strong correlation here again in oligodendrocytes. So then we went on. We generated HDAC1/2 double knockout mice with by crossing HDAC1, HDAC2 flux mice with mice that express the tamoxifen-inducible CRE recombinase under the control of the PLP promoter. So that is active in oligodendrocyte, actually mature oligodendrocyte, actually also in Schwann cells. So we looked at this, at what happens at 14 days post-tamoxifen, where we have checked that the levels of HDAC2 are decreased and of HDAC1. So you can see here immunofluorescence in the double knockout of HDAC2 compared to control in the CC1 positive cells. So in mature oligodendrocytes, you see that's mostly gone. And you see the levels of SOX10 are very, very low too. And this is just the same, but by Western blot analysis. And then we did the IP of SOX10 in the double knockout as compared to the control. And then we could see also that the increase co-immunoprecipitation of acetylated IF1A with SOX10. So very similar to what we have seen in the peripheral nervous system. So we have done other experiments to confirm the mechanism. So I don't show you all this. I will show you what we did with theophylline then. So we wanted to know whether theophylline could increase the levels of HDAC2, the remyelination, and so on. So for this, we used the lysoleciting model, which is a demyelinating, which induces a demyelinating focal demyelinating lesion in the spinal cord. So the advantage of this model is that you create a lesion that is localized, you know where it is. It induces fast demyelination and also nice remyelination. So it's quite a good model to study remyelination. We treated the animals with theophylline, same timing as in the PNS, so 4 days at 9 days post-lesion. And we started to look at 40 days post-lesion, and we also looked at 30 days post-lesion. So you can see here in this immunofluorescence that the levels of HDAC2 in the theophylline treated mice are really strong, as well as the levels of SOX10. You can see much stronger. And we had also much stronger myelin basic protein signal. So one of the main myelin proteins in the CNS. Then we did also electron microscopy. And here it was really amazing because we had really difficulty to find where the lesion was, right? Because we basically in all animals, so it's not that one was not well injected. So these animals regenerate really well. You can see that most axons are already remyelinated at 14 days post-lesion, and the remyelination is still better at 30 days post-lesion. And those young animals, they spontaneously can remyelinate in the CNS. So it's something that they can do. So of course, at some point, the vehicle-treated mice also recover. And then we looked also in the same mice in the unlesioned site to see whether theophylline has an effect on unlesioned regions of the spinal cord. And then we calculated the G ratio, so the thickness of myelin, and we could not see any effect. So theophylline doesn't increase the myelin thickness in outside of the lesion. So it's only in the cells that remyelinate that we have the effect. So then we thought, okay, we know that during aging, for example, in multiple sclerosis patients, the remyelination is a lot affected, and also during normal aging. So we decided to use to do the lesion, to use the model of demyelination, the lysoleciting model in old mice, and to do the treatment with theophylline. And here again, we could gain. We had a big advantage of remyelination, percentage of remyelinated axons in the lesion site. So potentially theophylline could be efficient to promote remyelination in young adults, but also in old individuals. Yeah. So as a conclusion, so I didn't show you everything, but as a conclusion, we could show that we have a similar mechanism in the PNS and CNS with the interaction of acetylated IF1A1 with SOX10, the deacetylation with HDAC2. We even have shown that there's a TIP60 that is involved, and that we can promote remyelination by theophylline by increasing the levels of HDAC1/2 or of HDAC2, sorry, which then will promote the relocalization of IF1A1 in the cytoplasm and maintain SOX10 on this target gene. So yes, so I'm done. So what I showed you was mainly the work of two fantastic PhD students in my group, Adrienne Vaquier and Mer Duman. Valérie Brugger did also some previous work that was great also on the function of HDAC1/2 after a lesion. So she also did that work. So they all moved on now, my fantastic student, but now I have other fantastic students. So it's okay, we're not, yeah. we're not left alone. So Nadej Herzog and Gianluigi Nocera are also very good PhD student. And I moved recently my lab from Switzerland to Germany. So we almost set up now, almost fully set up. So yeah, so that's great. And those are our collaborators and all our funding.